Have you ever stopped to think about how scientists can actually read the code of life? I mean, it's completely invisible. Well, today we're going to open up the molecular biologist toolkit and check out the seriously ingenious techniques they use to isolate, identify, and sequence our DNA.
So right off the bat, here's the fundamental problem. Inside every single one of your cells, you've got this chaotic, microscopic soup of DNA, RNA, and proteins, all just tangled together. If you want to study just one of those things, you first have to figure out how to sort through all that chaos and, see the invisible. So, first things first, the challenge is separation. How do you untangle that molecular mess? Before you can even think about analyzing a gene, you've got to somehow pull it out from everything else. And the answer is this brilliant technique called gel electrophoresis. Just think of it like a molecular sieve, or maybe even better, a microscopic racetrack. It is an absolute cornerstone of molecular biology, and the idea behind it is just so simple and elegant. So how does this little race actually work? Well, you start by loading your DNA samples into tiny wells in a slab of gel, which is kind of like a porous yellow. Now, here's the really clever part. DNA has a natural negative charge, so when you apply an electric current, all the DNA gets pulled toward the positive end. But the gel itself is like a maze. The smaller, lighter DNA fragments can zip through it no problem, while the big, bulky ones get all tangled up and move much, much more slowly. And the result? You get a perfect sorting by size, with all the smallest fragments racing the farthest down the gel. Then you add a dye that makes the DNA glow, and you see these distinct bands. What was once a totally invisible mess is now a clear, organized pattern. Pretty cool, right? Okay, so we've sorted our DNA into thousands of different fragments. But a lot of the time, that just looks like a long, blurry smear on the gel. So the next question is, how on earth do you find the one specific gene you're actually looking for in that massive genetic haystack? Well, this problem was cracked by a technique called Southern blotting. And yes, it was pioneered by a scientist named Edward Southern. It's an incredibly clever way to transfer the DNA pattern from that flimsy gel onto a much more durable membrane, which basically sets the stage for a genetic search and rescue mission. So here's the plan. You create something called a probe, and you can think of it like a molecular bloodhound. It's a short piece of DNA that's designed to perfectly match the sequence of the gene you're hunting for, and it's tagged with a label that we can make glow. When you wash this probe over the membrane, it literally sniffs out and latches onto its one and only target, completely ignoring everything else. And just like that, the probe does its job. That overwhelming smear of thousands of DNA bands just vanishes, and a single glowing band appears, right where your gene is located. You've officially found the needle in the haystack. Now, this isn't just some obscure lab trick. This exact technique is the foundation of something we've all heard of, DNA fingerprinting. It's basically just Southern blotting, but applied in a way that completely revolutionized forensic science. So how does that work? Well, it turns out our genomes have these quirky regions called mini-satellites. They're just short DNA sequences that get repeated over and over again. And the amazing thing is, the number of repeats is dramatically different for almost every single person. So if you use a probe that targets these regions, the Southern blot creates a unique barcode of bands for each individual, a pattern as unique as an actual fingerprint. And the impact of this has been just undeniable. In forensic cases, scientists can compare the DNA pattern from crime scene evidence, say blood or hair, to the pattern from a suspect. A match can provide incredibly powerful evidence. I mean, it did in one of the earliest cases, where DNA from a crime scene was shown to be a perfect match to a suspect's DNA, and that helped secure the conviction. All right, so we've sorted the DNA and we found our gene, but now we're at the final and maybe the most ambitious challenge of all. How do you actually read it? How do you figure out the precise sequence of A's, T's, C's, and G's, the actual letters that make up its instructions? The huge breakthrough here was a Nobel Prize-winning method from a scientist named Frederick Sanger. It's called, appropriately, Sanger sequencing, and it's built on this absolutely ingenious concept known as chain termination. The logic behind this is just brilliant. In the lab, you make copies of your DNA, but you sneak in these special terminator bases. Whenever the copying process accidentally grabs one of these terminators, the process stops dead in its tracks. You do this in 4 separate batches, one for each letter, and what you end up with is a whole collection of DNA fragments of every possible length, each ending with a specific letter. Then you sort these fragments by size using gel electrophoresis again, and you can literally read the DNA sequence, one letter at a time, right up the gel. Now, at first, this was a painstaking manual process. It used radioactivity and required scientists to read these long X-ray films by eye. But then, technology took a massive leap forward with automation. Instead of 4 different lanes and radioactive tags, automated sequencers use 4 different fluorescent colors, one for each DNA base. All the fragments run down a single tiny tube, and a laser scans them as they pass, reading the color of the final base on each fragment and feeding it straight to a computer.
And the scale of this is just staggering. A modern sequencing machine can accurately read over 200,000 DNA letters in a single 3-hour run. This incredible speed is what finally made huge projects like sequencing the entire human genome even possible. So, let's bring it all together. We've gone on this journey from a tangled mess of molecules all the way to a readable, letter-by-letter genetic code. And these aren't just abstract lab procedures, right? They are the fundamental tools that really unlocked the secrets of our own biology. So let's just do a quick recap of the toolkit. The problem was sorting a mix of molecules. The tool, gel electrophoresis. The problem was finding one specific gene in a massive crowd. The tool for that, Southern blotting. And finally, the problem was reading the gene's actual code. The tool, DNA sequencing. A specific, brilliant tool for every single one of those fundamental problems. You know, when you put it all together, this toolkit allows us to read the book of life. It lets us diagnose genetic diseases, understand our own evolution, and even engineer new biological solutions. With this incredible power at our fingertips, the only real question left is, what chapter should we explore next?